Hello, folks. In this video, we're going to cover different muscle fiber types. So we've learned all about muscle fibers in general, their anatomy, their physiology, what substrates they use to make ATP, where ATP is used during a muscle contraction. So we've now come to this point where we have to talk about these different types of muscle fibers. So this is going to cover the last set of objectives in our learning outcomes here, or the last set of outcomes, basically 15 through 18, right? So hopefully by the end of this lecture, you'll be able to predict whether different muscle fiber types are going to behave primarily aerobically or anaerobically based on their anatomy, that you'll be able to distinguish between the primary type of metabolism that they do or that they perform, again, based on their anatomy, which type of molecules they'll be using to make fuel, and during what type of activity or what intensity of activity they're going to be most active. And then we will definitely compare the sort of the anatomy of them, both the sort of microscopic anatomy and then intracellular anatomy. And then we're going to talk about how they can both be not remodeled, but they can both adapt to the type of activity that you primarily ask them to do. And so we'll be doing that 15 through 18 in this lecture. Okay, so muscle fiber types. This is my favorite part of of muscles, I think. Although I like the whole origin, insertion, and action, especially origin insertion, because it tells you something about the action. But this is definitely the coolest part, I think, about muscles and muscle physiology. Okay, so let's take a look at these different fiber types. Whoops, what happened there? All right, so muscle fiber types, we talk about sort of the polar opposite types of fibers, but it turns out there's a whole gradation of fibers. So it represents a continuum rather than two different types or even three discrete types, which is kind of how your book presents it. There's really a continuum of different types of muscle fibers. We often have talked about in the past, and it's worthwhile thinking about the poles so that we can describe their internal anatomy and therefore predict their physiology. So there are slow twitch fibers. Slow twitch fibers we call type 1 fibers. So slow twitch, you may also hear them called red muscle. This is really referring to the dominant fiber type in a whole muscle rather than an individual muscle fiber. But it turns out that really muscles are made of a mix of fiber types. And type 1, is the type that is the slowest to contract and relax. So this is why we call it a slow twitch fiber, because in a single twitch contraction, this type is the slowest to build up to peak contraction and get back to relaxation, to complete relaxation. So these are pretty small in diameter, meaning they don't have a lot of myofibrils, and within the myofibrils, they don't have a ton of myosin. They have a really slow contraction phase, right? So they're slow to reach peak contraction. They're loaded with mitochondria. So intracellularly, these are all sort of different things, right? I mean, this is like something you'd see at the level of looking at it at a microscope. This is something you would actually measure, right? The physiology of the contraction speed. This is something you would look at sort of cellular level, right? The number of mitochondria. There's lots of mitochondria in these. They are heavily vascularized. So this is, again, sort of a microscopic level, but at the level of the whole cell, right? Because you remember the places where we see vascularization within a whole muscle is in two different layers of connective tissue. So if you remember way back to the 3 layers of connective tissue, the one that goes around the whole muscle, the one that surrounds fascicles, and the one that surrounds individual muscle fibers, hopefully you remember that we see vascularity, capillaries, and blood vessels, larger blood vessels, in the endomysium, that inner layer, and the epimysium. Is that right? Yes, the layer surrounding fascicles. So it's heavily vascularized, really good blood supply. It's relatively weak. These, when we give them, when physiologists challenge these type 1 fibers with a weight and ask, let's contract it, and can you move this weight? It turns out they're relatively weak compared to other fibers. And they have lots of myoglobin. You hopefully, or maybe you haven't, but it does heard of hemoglobin before. Hemoglobin is basically a protein that you have embedded in the cell membrane and then circulating within the cytoplasm of all of your red blood cells. And so you may have heard of it. You may know that red blood cells are responsible for carrying oxygen. And hemoglobin is the molecule that actually binds to oxygen. Well, myoglobin is a very close cousin to hemoglobin. It differs in structure, and it resides not in red blood cells, but within muscle tissue, right? So this is embedded within muscle tissue and in the sarcoplasm of muscle tissue. Thus the name myo, right, in reference to myofibril, myofilament, in reference to muscle cells, right? So there's lots of myoglobin here. This is basically a way to store oxygen on site within the cell. And you know how important oxygen is to fuel aerobic respiration. So because of all of this, and something we'll talk about in a minute, the small diameter, the slow contraction phase, and it being relatively weak, that has to do with the mechanics of the actin and myosin during a twitch contraction. But the rest of this is all about how does this muscle uh, make its energy primarily? In what way is it really good at making energy? And hopefully you can see with a ton of mitochondria, a lot of vascularization, so a lot of blood moving nutrients in and carrying waste away, and a lot of myoglobin, which binds to and stores oxygen. This type of muscle is incredibly good at aerobic respiration. It generates ATP best in an aerobic fashion because it's set up to get lots of oxygen delivered, to then process, to feed electrons into the transport chain of the mitochondria and make ATP aerobically. And when it runs out or is not getting enough via vascularization, it's got the stored oxygen to act as that final electron receptor in the oxygen transport chain. So these muscles really behave aerobically. They tend to, because of their slow contraction phase and they tend and relaxation phase, they tend to be most active during periods of endurance type of exercise. So moderate endurance exercise. They can produce produce ATP very efficiently over a long period of time because of these mitochondria vascularization and myoglobin. So they're very good at behaving aerobically. Just think about going for a jog, think about going for a long swim across Lake Washington and back, think about riding your bike, 15, 20 miles or more. A leisurely activity, sort of. These are type 1 fibers are primarily active and they're really good at sustaining that type of activity. Okay, the other end is fast twitch fibers. And there are two varieties. There's type 2A and type 2X. It's also called type 2B. You'll see it differently, expressed differently, but these are the same, 2X, 2B, and then 2A. So these are fast twitch fibers. This is slower than this type, right? So this is the fastest type of fiber. So what are these like? These have a large diameter, right? They have a lot of myofibrils and a lot of myosin heads within those myofibrils. They have a fairly rapid contraction phase, about 5 to 10 times faster than slow twitch. So about 5 times faster and about 10 times faster than slow twitch fibers. So they're very good at contracting very quickly, right? So you should be thinking to yourself, okay, large diameter, probably pretty strong, stronger than these anyway, because they got more myosin. You should be thinking these are probably active during very rapid movements, right? So sprints, because they contract a lot faster. That means the muscles can contract and relax and reset and be ready for another contraction quite a bit faster. They have few mitochondria, right? So they don't have a lot of the machinery to make ATP aerobically. they're poorly vascularized, which is why they, I talked about this as sometimes being called a red fiber. These are often called white fibers because they lack the vascularization. And so under the microscope, they don't have a lot of blood supply. So they're poorly vascularized. They're not getting rapid delivery and rapid delivery of nutrients and expulsion of waste products. They're about 3 times stronger than a type 1 fiber, right? So these are also they're great for short, intense bursts of activity, right? Think of lifting a really heavy weight, right? And they have very little myoglobin in contrast to these type 1s. So they're not storing very much oxygen within the muscle cell. So hopefully you can see this suite of characteristics means that these are not good at aerobic production of ATP. They're just not. They don't have the ability because they don't have many mitochondria. They don't have good vascularization. They don't store oxygen on site. They're really good at producing ATP via glycolysis. And so the one thing that I sort of neglected to add here are the enzymes responsible for glycolysis are very high in these, right? And relatively low in these type of fibers. So these actually have a lot of glycolytic enzymes, those glycolytic enzymes that they have, the genes for glycolytic enzymes are transcribed and translated at a much higher rate in these fast twitch fibers than they are in these slow twitch fibers. So there's a lot more of those proteins floating around. So yeah, these are great for rapid strength based activities like sprints, right? Okay, but that's not all, because there are hybrid types, right? There are fibers which are a hybrid between type 1 and type 2A, and type 2A and type 2X, or type 2A and type 2B, right? So there are hybrid fibers too. Okay, so what is this 1AXB? What is all that type 1, type 2? What is that referring to? It turns out this is the name we give, or the little number we give, that type 1, type 2A, type 2X, to the type of myosin that is transcribed and translated by that cell, the dominant type of myosin. So remember, all cells are the same in terms of their genetic material, their DNA, right? All of the cells of your body, except for your sex cells, are the same, right? So this just turns out that these fibers basically only transcribe and translate a myosin that's relatively slow. And these fibers only transcribe and translate a myosin that's relatively fast. And these hybrid fibers can switch. So they transcribe both type 1, for example, a hybrid type 1 2A fiber would transcribe and translate both type 1 myosin and type 2A myosin, or type 2A myosin and type 2X myosin. So this allows some flexibility, right? Having a bunch of hybrid fibers allows you to change the dominant myosin fiber that you produce, sort of based on the activity that you primarily do with that muscle that has that hybrid fiber in it, right? Okay, so I'll just show you some data that supports what we've just been looking at, right? So this is looking at myosin fiber type 1, 2A, 2X, and then these hybrid types. So here is the force that's produced. This is as a percentage. Here's the power. Remember, this is the ability to move some mass over some length, right? So over this total power output, what this curve is basically showing is the total power output, right? This is the total force that's produced over the entire contraction phase of this muscle. Really contraction and relaxation phase, because right, even in relaxation, there's some tension being produced until we get to complete relaxation. So this curve is showing the total force that's produced, right? Think about it like that. This power is showing kind of how much power is produced at a given place during this contraction relaxation phase. So first, you'll notice that type 1 compared to like this hybrid fiber type, there's a not vanishingly, but a very, very small amount of force produced here, total force over this whole twitch versus over this whole twitch. Look at how much, right, we've got there. And the peak in the force is it comes up kind of the same spot, but look, it's really sustained for quite a long period. It doesn't drop that much over this slow contracting fiber versus this fast contracting fiber. It gets up there, peak tension very quickly, right? In terms of the power that it's producing, the power peaks very early and then it drops off really precipitously, right? So this is just kind of demonstrating weak, but producing more or less the same amount of power over the whole contraction. This extremely strong, but producing most of its power early in the contraction. So it's rapid production of a lot of power and then a steep drop off. Okay, so this has a really large force per unit area of this, really, and that's kind of another way of describing power. And then this fast rate of shortening, right? We get, and I'll show you that here in a minute. So this is kind of showing this incredible power and how rapidly it's produced in these fast twitch fibers versus these slow twitch fibers. And then this one is showing how rapidly they contract, where here this is MHC's myosin heavy chain. That just refers to the globular head of myosin, right? And we said that's what makes these fibers type 1 or 2 or whatever. It's the type of myosin they transcribe and translate. So here's our shortening velocity. This is how quickly a muscle shortens, right? This is then this is showing the different type of fiber. So, and then on this axis, this is showing the velocity of shortening in relation to how quickly a type 1 shortens, right? So this is just itself, right? So you can see it doesn't shorten very quickly, right? Here's a type 1 2A. It shortens about twice as fast as this type 1, right? If we're thinking about the shortening velocity in comparison to this, right? So this is somewhere at, well, this is at 1, right? That's about 1, right? This is at 1. And this is at about 2, maybe a little bit more than 2, right? This one's at 5, this type 2A. So in comparison to how fast a type 1 shortens, this is about 5 times as fast, right? That's the ratio of type 1, the shortening velocity of this focal fiber in comparison to the shortening velocity of this type 1. It's about 5 times as fast. It's about 8, 9 times as fast, right? And then this is about 10 times as fast. You can see as we move up here, and did it say what muscle we're looking at here? Yeah. This is just looking at the vastus lateralis muscle. So one of your large leg muscles, one of the large extensors of the lower leg, right? You can see there's that the estimate of this average, which is what this top bar represents, right? The estimate of this average really increases as we get into these hybrid fiber and even to this pure 2X fiber. There's quite a lot of variability in really how fast this contracts in comparison to type 1, but it's safe to say it's about 10 times as fast for this pure type 2X fiber. So this data is showing, right, that these different fibers, not only do they produce a different amount of force and do it at a different rate, but they also contract a lot faster than this type 1 fiber, up to about 10 times as fast on average. Okay. Now this is showing you the myograms for these muscle fibers. I take that back. This is showing you myograms of whole muscles. But you could imagine, and we talked about this, you could do a myogram for a single muscle fiber too. But this is looking at whole muscles. Now, hopefully you can see this extraocular muscle of the eye, the gastrocnemius. You remember this is that your calf muscle, the outer calf muscle, the most superficial calf muscle, the one that makes that little kind of U in the back of your leg. And then the soleus is kind of a broader, flatter muscle that's deep to the gastrocnemius, also in your lower leg. Right? So remember, whole muscles have all these different fiber types in them. It's just which type they express primarily that determines at least this contraction, this myogram, right? So what I hope you can see here now, first of all, what are we looking at here on the X and Y axis? So something you've seen before, tension, but look at this one is looking at the percentage of tension, percentage of the maximum tension it could produce. So while, you would say, you may look at this and say, wait, I thought, fast fibers or fast muscles were also stronger. They are. So if you were to actually draw this as a percent, or not as a percent of maximum tension, but just absolute maximum tension, you would expect these fast muscles to be way up here in terms of the maximum tension they could produce. But this is just looking at a percentage of maximum tension, because all we're really focused on here is looking at the duration of the contraction relaxation phases. Okay. So the soleus, when you look at this, you hopefully will be able to say, well, the soleus must be filled with mostly type 1 and type 1,2A fibers, because its contraction phase is the slowest of any of these muscles, and its relaxation phase is the slowest. It takes, on average, about 100 or about 200 milliseconds to complete a total contraction and relaxation phase, to complete one twitch contraction of the muscles, fibers of the soleus muscle. It takes a pretty long time. In comparison to the gastrocnemius, this must have many more hybrid fibers and type 2A or 2X fibers, because this reaches its contraction phase much quicker, and the relaxation phase is much shorter. This isn't even 80 milliseconds for a complete twitch contraction of the fibers of the gastrocnemius muscle. And then looking at this extraocular muscle of the eye, right? And of course, you can think, I'll just move my eye around, right? I'm just moving it around. You can see how fast that is, right? How quickly the eye moves around when I use those extrinsic eye muscles. So this must be loaded with type 2A X and type 2X muscle fibers, because it's an incredibly fast contraction and relaxation phase. They're on there at like 20 milliseconds. Yeah, about 20 milliseconds for that to complete. Okay, so when you look at this, I want you to be able to say, if you were to not label these at all, that you could say, hey, this is either, this is a slow fiber, type 1, maybe type 1,2A, or it's a muscle that is full of relatively slow fibers, and that this one is a muscle that's full of relatively fast fibers, or if this is an individual muscle fiber, that this is a muscle fiber full of fast, type 2AX and 2X fast fibers, right? Okay, and this is just to show you the sort of microscopic view of these and what that looks like. This is clearly, or this must be, a muscle with a mix of fibers, right? So perhaps this is type 1 and this is type 2A. They've labeled this as R for red and white. You can see a capillary here next to this red fiber. Here's another. Here's one small capillary next to this white fiber. Well, what do we see here? It's dark. It's small diameter, right? So it's got good blood supply. It's got a lot of myoglobin. It's relatively weak because it doesn't have a lot of myofibrils or myosin in it. This is great at producing ATP aerobically. This is an aerobic type of fiber. Here they must have found this is clearly a different muscle that is almost entirely type 1 fibers. Look at how the diameter is pretty much invariable here. But here's a much thicker fiber. Perhaps this is type 2A or maybe even type 2X. But it's much paler because it doesn't have a good vascular supply and it doesn't have a lot of myoglobin to store oxygen. It has a lot of glycolytic proteins. It's a large diameter, which means it's got a lot of myofibrils and a lot of myosin within those myofibrils. Right? These are easily fatigued. Right? These get tired fast because they're great at producing a lot of tension quickly, but because they're not aerobic, they have a difficult time maintaining their ATP production because they have to do it all glycolytically. And remember, that's super inefficient in terms of the total breakdown of glucose. Here in this top, this would net us like 36 to 38 glucose or ATP in total from glycolysis and the Krebs cycle and oxidative phosphorylation in the mitochondria. And this is going to net us 2 ATP for every glucose because it's terrible really at doing that aerobic respiration. Okay. So now let's think about how muscles can adapt, because we do have all these hybrid muscle types, right? And you'll read about this in that article that I assigned to you, genes and what is it called? I can't remember, but it's about how your genes are turned on when you exercise, and which genes are turned on in response to different types of exercises. So muscles can make adaptations, and they can make fairly rapid adaptations and fairly long-term adaptations. So rapid adaptations and long-term adaptations. The rapid adaptations have to do with the transcription of genes that operate very quickly in the cytoplasm and on the plasma membrane of the cell. These long-term adaptations are much more about structural reorientations, structural changes, additions of big proteins like myosin, repair of damaged tissue, and we do have a type of cell called a satellite cell that exists around muscle tissue, and there are many around all of our individual muscle cells. And these satellite cells repair that muscle when it does get damaged in the course of normal use. And so this is all in the article. You can read about that. Here, I'll just show you what a chunk of muscle looks like before and after some intense exercise. This is actually taken from looking at older individuals trying to build strength and build muscle. But the same thing happens in individuals of any age. This is before exercise. You can see these beautiful little sarcomeres here, right? In this myofilament that's stretching this way and this way. You see these great myofilaments all arranged neatly, sarcomeres all in a line. Great. Now, after some intense exercise, they did some resistance training. So they were lifting weights to failure, right? This is what the same region of the muscle looked like, right? Hopefully you can see this is a mess now. Look what happened to this beautiful sarcomere and this myofilament. It's totally scrambled, right? And all these, they're scrambled, they're broken, right? That's what happens when you stress muscles to the point of failure. When you stress them to the point of fatigue that doesn't allow you to continue the exercise, and you're doing it with sort of weight, so you're doing some kind of resistance exercise, this is what you get. You break your myofibrils, you break the myofilaments. So the actin and myosin that are trying to resist the forces or the force of the weight that this individual was lifting broke in the process. That's just what happens. And this is the only way that muscle rebuilds itself larger and stronger. You have to break the myofilaments. And just like a bone, and we saw that when a bone heals, initially that healed portion, that callus, is thicker and stronger than the rest of the bone. The same thing happens with muscle. This muscle has just experienced force. You've just challenged it in a way that caused it to break, right? To fall apart. Right? So the muscle responds by rebuilding itself stronger, by adding myofilaments and by adding myosin to the myofilaments. Now when that happens, when you do this to a muscle repeatedly, and it starts to add myofilaments and add myosin to those myofilaments, that's when we get increases in muscle size, which is a long-term adaptation, and that's called hypertrophy, a growth in muscle. Okay. This is just showing you some satellite cells, right? In the process of the formation of a mature muscle, it actually starts with the fusion of dozens to hundreds of individual myoblasts during development that become the skeletal muscle, and that's why it's multinucleate. But you always have satellite cells hanging out on the outside. Here's our satellite cells. And when we do that to a muscle, when we just trash its sarcomeres and break them, right, then these satellite cells actually differentiate and start producing products to repair those muscle cells, and they merge with this muscle cell at sites of damage to help to repair it. Okay. Now let's think more about these adaptations, the rapid adaptations and then the long-term adaptations. So when you challenge a muscle cell, especially aerobically, right? Especially an aerobic challenge, so you're doing some endurance exercise, you get some pretty rapid adaptations. And even when this verges on the endurance to kind of resistance training, right? So even when it verges on becoming more anaerobic, right? And even when you start relying more on glycolytic products from your muscles that are active, they will all respond in this way. They'll increase their glycogen storage. So after this challenge that you give them, they're going to store more glycogen than they had before. They're going to increase their ATP and creatine phosphate storage, right? The more of this they have on hand, the longer they can sustain activity. They're going to increase their glucose transport proteins. So of course, those glucose transport proteins are import proteins, right? They're bringing glucose in from the vasculature, from the bloodstream. So you increase your glucose transport proteins, your import proteins of glucose, so you can more rapidly take it up from the blood and convert it to ATP. You also increase your lactate export proteins, right? So remember, when you've challenged a muscle cell so much that it can no longer sustain its normal activity via aerobic respiration, and it has to switch to more of glycolysis, that's when you start getting a buildup of lactate proteins. We got to get rid of those so we can take them to the liver, rebuild it into glucose, blah, 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 right? So you increase your lactate export proteins. Another one that increases, which I actually neglected here, but it is an important one, is your hydrogen ion shuttling proteins. So remember that when we start operating more anaerobically and we produce more lactate and we're not able to do as much aerobic respiration, we also increase our hydrogen ion production, right? And so we got to get that out of there, because that induces metabolic acidosis, which leads to muscle fatigue, right? So that approaching fatigue stimulates our muscle cells to produce more hydrogen ion export proteins as well. The other thing that happens, the other thing that happens is increased neuromuscular coordination that happens pretty rapidly. And in fact, if you start lifting weights or doing some kind of strength training program, the first increases in strength are not due to muscle growth in size or hypertrophy. It's due to increased neuromuscular coordination. So improved communication and efficient communication between the motor neuron and the individual muscle fibers that it contacts, so that you end up getting you basically end up getting all fibers, whether they're endurance fibers like aerobic fibers or glycolytic fibers, being more efficient at firing just the right number and just the right of motor units, or just the right size of motor units to lift whatever weight you're lifting, but not overdoing it. And that leads to you being able to sustain that activity for a longer period of time and lift a weight for a longer period of time. Then there are long-term adaptations. These typically take on the order of weeks, right? Where these rapid adaptations are taking place immediately after a single bout of exercise. You're getting these rapid adaptations. These are more like a week long or longer. You get an increase in mitochondrial density. Mitochondria get the signal that there aren't enough, they're not producing ATP rapidly enough. So we just increase their density. Remember, these are these mitochondria, having come from proteobacteria that were ingested long ago by a sort of proto-heterotroph, or proto-eukaryote. These things are not reliant on the cell cycle in order to replicate themselves. They replicate themselves via fission whenever there's the need, right? So these can increase in density over a period of many days to weeks. You also increase your capillary density. So your cells secrete angiogenesis factor, which actually attracts capillaries to the muscle cell. So more capillaries invade the endo and epimysium and contact that muscle cell. And then there's hypertrophy, which I talked about before. This is an increase in the size of muscle fibers due to an increase in the number of myofibrils and the number of myosin filaments within those myofibrils. And it turns out, to do this, you really have to exhaustively stimulate your muscle. And that, we used to think that was by lifting heavy weights to exhaustion, but it turns out you can do it, although the gains may not be as significant if you lift moderate weight to exhaustion. So rather than lifting something 10 times until your muscle fails, you might lift it a lower weight 30 times until your muscle fails. But as long as you get to exhaustion, you should be stimulating hypertrophy. And this is the actual growth in size of a muscle, simply by the addition of myofibrils and myosin filaments. Right? Okay, finally, physical conditioning or training. When you train aerobically and you're just taxing the aerobic fibers, you don't get a lot of change in your type 2, pure type 2 fibers, type 2A, pure type 2X. But you do get changes in those hybrid fiber types, the 1, 2A, and 2A, the 1, 2A primarily, right? These get the signal that they need to produce more ATP when you tax them aerobically. And so, and aerobic endurance simply means muscle activity that's supported by mitochondrial activity alone, rather than shifting to glycolytic activity. So given that this aerobic activity is affected by mitochondrial density, capillary density, myoglobin concentration, ability to shuttle to export lactate, these are the types of changes you're likely to see if you do a lot of aerobic or endurance training. But when you start doing anaerobic endurance training, that's when you're going past aerobic and you're shifting to muscle activity that requires not just mitochondrial activity, but also glycolysis, continued glycolysis in the absence of additional mitochondrial activity. That's called your lactate threshold. And this is affected by, of course, all the things that you just need for glycolysis, like your ATP and creatine phosphate stores, or the things you need for the glycolysis, or the things you need to generate ATP quickly. So if you just increase your stores of ATP and creatine phosphate, you're increasing your anaerobic endurance, right? Because these are stored products. You're not doing anything really to use them. Increasing your glucose stores will affect it, because this gives you more substrate to do glycolysis on, right? You can also other things that happen are you increase your tolerance to acidity, primarily by increasing the number of hydrogen ion pumps, export pumps. So you increase your tolerance to that acidity. And I'm blanking on something else for anaerobic endurance. Well, your hybrid fibers, type 1, 2A, will now produce more type 2A fibers, right? Instead of type 1 fibers. If you have type 2A, 2X fibers, they'll probably shift to making more type 2X fibers, right? And the other thing is you increase your production of your glycolytic enzymes, right? So you increase that's just the concentration of the glycolytic enzymes you have in the sarcoplasm. So these are all different changes that happen in response to aerobic endurance training and anaerobic training. Right. Okay, that was a long one. Please let me know if you have any questions about any of this fiber type stuff, the physiology of these muscle fibers, the anatomy that underlies those physiological differences, or anything about aerobic or anaerobic endurance and the adaptations that muscles can undergo or can achieve when they are stressed, primarily aerobically or primarily anaerobically. Okay. See you again in a future video.